Oxygen, a basic necessity. Without it, our body metabolism fails to produce energy. Normal activities require normal levels of oxygen under normal body conditions, known as normoxia. If we go uphill or do other activities which require more oxygen, then the oxygen level in the body decreases. This condition is called hypoxia. Hypoxia is a condition that has to be dealt with. But how? In hypoxia condition, our kidney cells produce the hormone erythropoietin, which then goes into the bone marrow to stimulate the production of more RBCs, which in turn trap more oxygen in their hemoglobin, which is the requirement. But erythropoietin is not required in normoxia conditions. But how does our kidney cells know whether we are in normoxia or in hypoxia conditions? For this, we have to look at the erythropoietin gene, which is responsible for the production of erythropoietin. Gene sends the encoded information through the mRNA by a process called the transcription. The mRNA then travels to the ribosomes, which converts it into the erythropoietin protein by a process called translation. The proteins controlling the transcription of the genes in the eukaryotes are called transcription factors. A cell has many such transcription factors. Further studies on a genetically altered mice revealed that a specific gene near the EPO, which was the hypoxia response element, was also responsible for the oxygen-dependent sensing. Its transcription factors were the HIF, which composed of ARNT, which was always bound to the gene, and HIF1 alpha, which was not. In normoxia condition, there is a presence of plenty of oxygen near the cell, which get converted into hydroxyl groups, which then get attached to the proline residue sites of a specific region of HIF1 alpha known as the oxygen demand domain. The proline residue sites are the open protein end acting as active sites. This type of protein modification is known as the prolyl hydroxylation. Prolyl hydroxylation helps a complex containing ubiquitin to bind to the hydroxyl groups, leaving HIF1 alpha unstable and vulnerable to the proteasome, which degrades it through proteolysis or peptide bond breaking. To which part was the complex bonded and why? The VHL gene was the answer. Research on von Hippel-Lindau disorder, a type of cancer, showed that a non-functional VHL gene created a condition of hypoxia by expressing hypoxia-regulated genes. A functional VHL gene could stop the cancer. So, a functional VHL is responsible for the oxygen-dependent degradation of HIF1 alpha. It is the VHL protein which gets itself attached to the hydroxyl groups. In hypoxic condition, as there is presence of no oxygen, then the HIF1 alpha remains stable with the oxygen demand domain and thus binds to the HRE gene, thus turning on the erythropoietin machinery, which then facilitates RBC production. For this, the Nobel Prize last year went to. This understanding may pave a way for the treatment of cancer.